All right, this is going to be a video over patterns of inheritance. This is going to cover the father of genetics, which is going to be Gregor Mendel. So when we talk about ideas of hereditary, before the 18th and 19th century, there was not a lot of information about hereditary. So we talked about botanists. So botanists are scientists that study plants. So when we say early ideas of hereditary, this would be when we were studying plants in the 18th and 19th century. So botanist, scientists that study plants in the 18th and 19th century. Produced hybrid plants. Now, what they mean by hybrid plants is they would cross plants basically of the same species, and they would get hybrids which looked a little bit different. Hybrids are plants that have been crossed with each other. For example, you could take a tall plant of the same species and cross it with the short plant of the same type of plant. So think of pea plants. That's what Gregor Mendel used, and they created hybrids. Now the hybrids are going to be a little bit varied from their parent plants for what we talk about in terms of strains. They do use hybrids a lot for different kinds of plants. We can cross different plants to create something completely different. So this is how we got our early ideas of hereditary, and Gregor Mendel is what we call the father of genetics. He was an Austrian monk, and he studied pea plants. So when we say Gregor Mendel, he is going to be considered the father of genetics. And everything that we've learned about inheritance and genetics comes from Gregor Mendel. He studied pea plants, and pea plants are really easy to study because they have a lot of characteristics that we can look at to make hybrids. So, couple of things. Pea plants can have purple flowers. So he crossed purple flower plants. So he made hybrids. And there's also going to be white flowered plants. So their flowers are different color. Now their seed colors, they had yellow seeds. Some of them had yellow seeds. And some of them are going to have green seeds. The seed shape, some of them had round seeds, and some of them had wrinkled seeds. So the reason he chose pea plants is because he could create these hybrids. Now we know that peas create pods, so some of them had inflated pods. Inflated means they're large, they're nice and thick. Some of them had constricted pods. If I were going to describe this in terms of a pea, a pod, I can't draw very good, but an inflated pod looks more like a green bean, where a constricted pod looks like you can see the peas inside of it. That's what the pods are. Now, the pod color varies in pea plants. You can have green pods. They also could be yellow pods. Now, they can be tall, or they could be short. There was so many different variations that Gregor Mendel could choose to create hybrids. So he chose to study pea plants because there's so many different pea varieties available. They're easy to grow, they're easy to self-fertilize, but you can also cross-fertilize plants to create hybrids. So we're going to talk about Gregor Mendel's, basically his experiment method. All right, the first thing that he did was he took his peas and he produced true breeding plants. Now, the word true breeding is something that we need to focus on. Something that is considered to be true breeding when you self-fertilize this plant. So if you take a tall plant, every time you self-fertilize or you produce another plant from one plant, it's going to remain tall. So he created true breeding strains. A true breeding strain is basically every time they produce a baby, they're always going to have the same characteristics or the same trait. The second thing is he cross-fertilized, and when you cross-fertilize, this is how you make hybrids. So let's just say he took a tall plant that every time it makes its own babies, it's going to make tall pea plants. And when you take a short plant, every time you take a short plant and it self-fertilizes, it makes a short plant. So what he did was he cross-fertilized. When you cross-fertilize, you cross-fertilize true breeding plants. These have to have alternate forms. So the example I'm using is a true breeding strain that we can say is we're going to cross a tall plant. That's a true breeding strain with a short plant. So this is going to be a tall pea plant and this one's going to be a short pea plant. Now both of these, when they're self-fertilized, these are going to be true breeding strains. So what he did was he crossed them. Now when we do crosses, you can do a test cross on a Punnett Square. A Punnett Square is going to help you figure out their babies or the generations that's produced. So he took a tall plant, which you would write at the top, and he took a short plant, which you're going to write at the side, and he crossed them. So what he did was he cross-fertilized. So this pea plant is going to cross-fertilize with this pea plant. So the way that you take your Punnett Square is you always put your big letter first and then your little letter second. So these in the box would be after you cross-fertilize. This is the cross-fertilize, these are new plants. So we took two true breeding strains that have alternate forms, tall versus short. So this is what we're crossing when we cross-fertilize. So the X represents that you're crossing a tall plant versus a short plant. They're cross-fertilized. And then the babies, or the offspring that are produced, are called the hybrid offsprings. And the big T and little t represent their genetics. So the third thing is he took the hybrid offspring. That's the babies created by true breeding. And he allowed them to self-fertilize. Now, self-fertilize is when you cross your own plant together. So self-fertilize you cross it with itself, and it created its own offspring as well that looked different than the parents. So this is the offspring of self-fertilization. So this was his experiment design. Now I'm going to go through his experiment design again, but I just want to talk about true breeding strains for a moment. If I took a tall plant, which is true breeding, every time it's going to create the same exact genetics. It has two of the same exact genes. So when you self-fertilize a true breeding strain, so we're doing self-fertilize, every time it's going to look the same as the original plant. That is what we mean by true breeding. So the first thing that Mr. Mendel did was he figured out that every time I use this one plant, it's going to look the same no matter what. So then he crossed two of his true breeding strains, and then he took those offspring and he let them self-fertilize to see what they would produce. And you can see that inside of the Punnett Square, they look a little bit different. So we're going to talk about different types of crosses and different types of dominants using Mr. Mendel's experiment design. So the first thing I'm going to talk about is monohybrid crosses and simple dominants. All right, when you use the word mono means one trait. So we're looking at tall versus short. We're also going to call this a simple dominance. There's different types of dominance. Simple dominance means that you're going to have genes that are basically dominant over the others. So when we say we have a monohybrid cross, this is a cross that studies only two variations. Of a trait. Now, what I mean by that is Gregor Mendel is only going to be looking at either how tall they are, or he only looked at their seed colors, or their round seeds versus wrinkle seeds. He's only using one characteristic. So peas can either have purple flowers or white flowers. That's one characteristic, is color of flowers. Seed colors is one characteristic. So he's only looking at one thing when we talked about the monohybrid cross. Now, because peas can have purple flowers and round seeds, if you did a cross that involved two things, that would be a dihybrid cross, but we're using a monohybrid cross. So it's important to understand that Mendel produced true breeding strains for 7 different traits in pea plants. So those 7 different traits that we talked about, this is only 6 of them, would be flower color, seed shape, pod shape, color of pods, tall plants versus short plants. Another one is flower position, which I'm not going to go into. Basically, he made true breeding plants for each one of these. So you could basically designate letters to represent each of these. For example, we used tall, which I'm going to use this one, and short. These are true breeding plants. So he used 7 different traits, and what he did was he was going to cross-fertilize them. That's part of his design. So he has 7 true breeding traits, and he started to cross-fertilize these, which is going to look like this. So, number 3, he cross-fertilized true breeding strains. Now, when you cross-fertilize true breeding strains, Mr. Mendel called this his filial generation. His filial generation is his first generation after crossbreeding. So we call this the F1 generation. When we take tall and we cross-fertilize it with a true breeding short, this is going to produce your F1 generation. The generation of hybrids. These are the offspring, the hybrid offspring produced by crossing two true breeding strains. So we do our cross. This is going to represent your F1 generation. You take your tall pea plant and you cross it with your short pea plant. You always put your large T in top. And when we look inside of these squares, we see that there is 4 chances of different types of traits. This is the F1 generation. There were 4 big T's, little t's. This is the F1 generation. Now, something that we noticed is none of these plants are short and none of them are medium. They were all tall. So when we say simple dominance, the large T is always dominant. So there were no plants with intermediate characteristics between the 2 plants. So what happened, this is simple dominance, all 4 of the F1 generation were considered to be tall because the big T is dominant to the little t. So all 4 of the F1 generation have a large T and a little t, but all 4 of them were tall, even though they have a big T and a little t. So there were no medium plants. These were all tall. So your F1 generation is your offspring that comes from breeding 2 true breeding plants. That is how you create your F1 generation. Now, your F2 generation. So what he did, third part of his design, was he took his F1 generation and then he is going to allow these to self-fertilize. The hybrid offspring, part of his experiment design, is the hybrid offspring were then allowed to self-fertilize. So we call this the F2 generation. The F2 generation. These are the hybrid offspring of 2 true breeding plants. were allowed to self-fertilize. So they cross with themselves. And this is going to create the F2 generation. So the F2 generation is going to be the offspring resulting from self-fertilization of the F1 generation. So let's see what that looks like. This is a Punnett square to represent self-fertilization. So the F1 generation is going to self-fertilize. So you draw your Punnett square. So you put one plant on top, big T, little t, big T, little t. Now you cross fertilize, but these are self-fertilization because they're the same. And then you put the letters inside of the square. So big T, little t, big T, little t. You always put the big T in top. Little t and little t. So these are representing the F2 generation. So you have one F generation that has a big T, big T. You have two of your F2 generation that has a big T, little t, and one of your F2 generation, which is little t, little t. This represents your F2 generation. Now, something was different in Gregor Mendel's pea plants that he noticed. These pea plants had different appearances. When we had the F1 generation, there was no intermediates. They were all tall. But he noticed that 3 of the plants were tall, and 1 of the plants was short. Now, we said before that if you had a large T, you had a tall characteristic, and this is why it's simple dominance. If you have a genetic trait that is dominant, then you are going to be that trait. So when we looked at these, this one right here, the 2 T's is obviously tall, and then the 2 that have a large T and a little t are also tall. So this is how we get the 3 that are tall. And then we had 1 that had 2 little t's, and that was the plant that was short. So this is where simple dominance comes in. When we say simple dominance, oh, that would be number 5. If you are having traits that are dominant, the form of the trait is expressed in the F1 generation, which we saw. In the F1 generation, we crossed a true breeding tall plant with a true breeding short plant. When the tall plant gave its genetic material to the short plant, we noticed that the tall plant was dominant. It had a dominant trait. Now, if something is considered to be recessive, the form of that trait is not seen in the F1 generation. We can go back to the same cross. The short plant gave its genetic material, so we would consider the little t the recessive form of the trait. So anytime we say dominant and recessive, we use big letters for dominance and little letters for recessive. So in the F1 generation, the dominant form of the trait was a little t. That's what's expressed. The recessive form is a little t. So going back to our F2 generation, our F2 generation exhibited both forms. It had tall and short. So we had 3 plants that were tall because these are dominant forms, and we had 1 that is short. So when we talk about the dominant recessive ratio, we basically would say this is a 3-to-1 ratio. So dominant to recessive ratio is looking at this. We say it is a 3-to-1. This is the dominant to recessive ratio. If you have a big letter, that is dominant. So we have 3 plants that are tall, 1 plant that is short, so we say only 1 has all recessive traits. Couple more definitions. Definitions that you'll need to understand. I'm going to change markers. We are on number 7. When we say the word gene, genes are in your genetics. This is going to be the information that is passed from your parents to yourself, or passed from parent to offspring. That is a gene. We pass genes to our offspring. Now, everybody has 2 sets of information from their own parents, but you can't pass both genetic information. That is called an allele. These are alternate forms of a gene. If we were talking about pea plants, basically you can say you have a dominant allele, a dominant allele would be represented by a large T. That's an alternate form. But you can also have recessive alleles. Recessive alleles are going to be represented by a little t. That's an alternate form, tall versus short. Now, we've learned that we have 2 sets of alleles. So if you have 2 of the same alleles, we say that those are basically homozygous. So we use different words to describe the alleles that you contain. When you use the word homozygous, homo means the same. This means you have 2 of the same alleles. When you use the word heterozygous, that means you have 2 different alleles. So, if you have 2 of the same alleles, that would be you have 2 dominant or you have 2 recessive. Those are homozygous dominant and homozygous recessive alleles. But if you have 2 different alleles, you have a homozygous, no, not homozygous, a dominant allele and you have a recessive allele. That's heterozygous in terms of letters. Now, we call this our genotype. What's in our genes? Remember that one gene comes from your parent, from your mother, and one gene comes from your mother. So we pass down genetic information, but we only give half of our information. But if we're talking about our genetic makeup, we call that our genotype. This is your total set of alleles from your parents. So you can have any different type of genotype. So because we're using the same letters, T, 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 when we talk about genotypes, you have to use the words dominant and recessive, and then you have to either represent if they're homozygous dominant or homozygous recessive, or if they're heterozygous. So my genotype, if we were looking at this, the genotype of this plant is heterozygous. It has a dominant allele and a recessive allele. But the F2 generation, it would have produced one plant that is homozygous dominant. It's got two of the same alleles, dominant, two big ones. It produced two heterozygous plants, so one dominant allele and one recessive. Heterozygous means two opposite. And it produced one homozygous recessive, which means little letters. Instead of saying you have one TT, two TT, and one TT, you have to start using the words dominant and recessive to represent the large letter and the little letter. And you have to say homozygous if they're two of the same, or heterozygous if they're two different. That's your genotype. So if you are or contain the two dominant alleles, your genotype would be called homozygous because you have two of the same, dominant. If you have one large one and one little one, they're two alternate forms, you're considered to be heterozygous. And heterozygous literally just means one dominant allele and one recessive allele. And if you have two little or two recessive alleles, homo means you have two of the same. You have a genotype that is called homozygous recessive. Now, this doesn't tell you the appearance. So when we talk about the outward physical appearance, you use the word phenotype. Phenotype, I want you to remember phenotype literally as the physical, PH, appearance. So this is the outward appearance of an individual based on the genotype. So, an example, if you are homozygous dominant genotype, your physical appearance would be that you're tall. That is your phenotype. You're a tall plant. If you are heterozygous, since we're doing simple dominance, you have one large T, your phenotype would also be tall because we're doing simple dominance. The dominant letter is over the recessive letter. But if you have two little letters, you're called homozygous recessive genotype, but your physical appearance is short. You're a short plant. That is your phenotypes. So you have to look at your genotypes to figure out your phenotypes. All right, now Mendel has two principles of segregation, principle of segregation, and he also has the principle of independent assortment. We're going to cover the principle of segmentation first. These are Mendel's principles. Your two alleles for a gene segregate during gamete formation. So what I mean during gamete formation is they're going to separate. If you remember in meiosis, to produce sex cells, your mother's and your father's genes separate during meiosis 1 when homologous chromosomes separate from each other. So during our gamete formation and meiosis, they're going to separate. And then they are rejoined at random one from each parent during fertilization. And if we remember fertilization, fertilization is when the sperm fuses with the egg. So a long time ago, we covered meiosis. And during meiosis, we learned that we could produce 4 different possible gametes. Here is my review of mitosis versus meiosis. Meiosis produces gametes. Your genetic material separated during meiosis. So your traits are going to separate. Now, we can use this information by drawing out the Punnett Square, which we've already been drawing. A Punnett Square is basically how you can cross a mom and a dad and show meiosis. So the father could be homozygous dominant. So literally, if you say you cross a man who's homozygous dominant, first of all, the gametes have to separate. You have genetic material. This is gene segregation during mitosis. This is the male. Those separate, so we divide them. Literally, this is a male that you're crossing with a female. So the gametes have to separate. So you could say that this is the female and her gametes have to separate during meiosis. Now they reform, they rejoin each other at random during fertilization. So the sperm cell has to fertilize the female cell. So what happens during fertilization is these rejoin together. So we use this Punnett Square to figure out what are the possibilities that could happen. So the male's gene from the sperm is going to randomly fuse the egg. This is the principle of segmentation. So literally, we are creating a new individual, mom and dad, and this is their possibilities of their offspring and their babies and their genotypes. So this is the principle of segmentation, and we're using a Punnett Square to help us figure this out. So these are the principles of segregation. I'm going to stop this video here because I want to start a new section on dihybrid crosses using two characteristics, and then I will give another principle called the principle of independent assortment.